Hello, this video unifies AGP glycoproteins and the proton pump of the plasma membrane with calcium signaling that regulates plant growth. Pembroke College has a great botanical tradition. Indeed, it is the Cambridge college closest to the Department of Plant Science. After years of effort, Li Tan and Masha Kerejewski's classical work resolved the formidable problem of AGP polysaccharide complexity, its repetitive consensus glycomodule of 15 sugar residues. High-resolution NMR techniques identified the precise glycosidic linkages. The galactan main chain, shown in dark blue, has two bifurcated side chains, shown in light blue, each with a penultimate glucuronic acid in red with a terminal rhamnose residue, and the calcium ion in green. This led directly to a 3D molecular model and molecular dynamic simulation of calcium binding. Extension of a pollen tube tip oscillates. Each growth cycle depends on a molecular turbine which generates proton jets. These eject calcium ions from the glycomodules. Listen to the molecular traffic noise at the plasma membrane that reflects its dynamic activity. 4 distinct phases of the growth oscillator, driven by the proton pump, are 1. Quiescent, 2. Auxin-activated, 3. Fully active, 4. Recovery. The video simulates these plasma membrane ion fluxes. Phase 1 of the pinball machine, quiescent. Minimal cell expansion, minimal cell wall stress, minimal calcium influx, and the lowest proton pump activity. Phase 2, activation. Cell expansion increases wall stress. Its transduction via hectian adhesion opens the calcium channels and initiates auxin binding that activates the proton pump. Phase 3, maximum activation. Stress vector points auxin efflux pin proteins towards the stress. Increased auxin levels maximize proton pump activity. Protons discharge glycomodule AGP calcium. Calcium channels generate cytosolic calcium waves that activate exocytosis of pin proteins and their dynamic relocation. Wall precursors reinforce wall and slow cell expansion. Phase 4. Return to quiescent phase. Stress relaxation of reinforced wall closes calcium channels. Auxin dissociates from proton pump, deactivating it. Cytosolic calcium recycles via AGP precursors and calcium transporters to replenish periplasmic AGP calcium.